So before we get started, I'm just going to say that the exam will be what is it, December 18, 19? It's on the Thursday that week, and it's here at 2 o'clock, and it's for 3 hours, and you can use your computers, the internet, any books you like uh, to solve the problems. Any questions about that? Yes. I don't know. How many problems? We don't know. I mean, some of them will be very easy and difficult. But yeah, no, it will be. I mean, the plan is that you should be able to finish it easily in 2 hours, and then you'll have an extra hour if you want to, or not. Yeah, and then you just submit it by email to the same email address, and the so next Tuesday for the tutorial, you'll bring your favorite signal or image, and then we'll discuss that. And then the Tuesday thereafter, we'll go through all the homework. And if you have any questions related to anything. Yes. Yeah. But I mean, it won't be. Yeah. I mean, you won't need to do a lot of programming, but you might need to take a script and modify it slightly. And in that case, it will probably be one of the scripts that are related to the homework. But it will be, you won't have to write a long new script, but some modifications, definitely. Sure, whatever you want. Yeah, but you should do the exam on your own, not send each other emails. Okay, so today we're going to talk about image informatics. And last time we talked about signal processing of one-dimensional signals. So this is just now expanding that to 2 dimensions instead. And a lot of the approaches are quite similar, but there are some approaches that are specific to images. And so the nice thing with images is that anyone can look at it and you can see it's nice, lots of colors, it's pretty. And compared to a one-dimensional signal, which is often considered a little bit more boring. But often the one-dimensional signal actually can give you more information. And we'll talk a little bit about that. So I'm going to talk a little bit about then going through, and to start with actually cutting a slice through an image and looking at it as a one-dimensional signal. A little bit about different ways to do thresholding, and then how one can find and characterize objects in an image, and then also how to classify some simpler structures. And then we'll get into doing a little bit of simple simulations to see if what we see of different particles overlapping is random. So an image in Python is simply a 2-dimensional numerical array. So we have rows and columns, and at each pixel there is a value of the intensity of that pixel. And that's sort of the basic element. So you're familiar with arrays. So it's actually very easy to to look at these images when inside of Python. So there are lots of useful packages. For all these things, you often don't have to write a lot of code. You can write one line to find all the properties of all the objects and things like that. So there's a lot of people have built a lot of packages that are very useful, and then you just need to reuse. And then we'll talk a little bit about that later. So the first thing, I thought I'd go through an example of a few gel images. So here we have 12 gels that are put together, and as you see that some of them are straight, but a lot of them are not. And so the idea, what we want to do is generate a lot of, to automatically analyze this. And the background is that this company, CDI Laboratories, are testing antibodies. So they generate hundreds of these gels, so they don't want to analyze it by hand. So the first step is we need to split it up into the individual gels. And one way of doing this, so we can then put all the 12 gels in a row. And if we just add up all the points on the y-axis at each x position, then we get a spectrum like this. So now we go from this image to a one-dimensional signal. So we see that we have these large peaks in the red channel, and most of these large peaks correspond to the marker lanes. And that's what we then can use to see where each of these gels start. So what we do is we find these peaks, but some of these large peaks are red peaks are not does not correspond to like we get quite large signal from this band, which is not a marker lane. So what we need to do after is to go through for each of these peaks, look at the distribution of intensities in the other direction. So here we have is one of the marker lanes that's been turned around. So then we see that we get these peaks in the red channel for the for each of the bands. So then a few different things can happen. So one thing is that, so these are two of the marker lanes that look quite good. So meaning that there's a strong red signal, almost no green signal. The intensities of most of the bands are similar, although it goes down a little bit at the low molecular markers. So that looks decent. But on the other hand, here we have a marker lane where this band here is almost invisible. And there is another band here that's also very weak. So we have, and these things happen all the time, even though they try to run this as consistently as possible. And so, but these, we want to be able to recognize these as marker bands, the three up here, while the bottom ones are not. So we see that here we have sometimes large green signals. We have, even the cases where we have a lot of red peaks, they don't have the right spacing. So we can write down a few criteria of what how to characterize. And the main thing is that we want the number of strong red bands for it to be considered a marker lane. We want the band intensity to be within a certain variation. We have to have a low green signal, and the relative band locations have to be consistent. So this we can then just add in as rules to decide, is this a marker lane or not. So the next step is that if we want to detect lanes, they're usually crooked. So what we want to do is to straighten them out also. Mainly because if they're straight, it's easier to analyze them. So one way to do this is to scan over the gel and sum up along a line, and then both move the position of the line and change the angle. And then find minima in this case. So here we would find that the intensity, when we write between these two lanes, that's the low. So then we can find what's the angle of that lane. And then we can straighten them out. And then, of course, if they're straight, it's much easier to analyze them. So then what we do is we separate it into the gel signal, and we can calibrate so that we put labels onto the marker lanes automatically, and we can also always make that the size of the image is the same every time. So we stretch them according to the marker lanes, and we can add other kinds of annotations. Then now that we have the gel size is standardized, we've straightened out the lanes. Now we can go in and actually integrate the different bands, so detect the bands and calculate what's the intensity, the area under, the volume under them. So then we end up with we can get a table that we can then use for other purposes. So now here what we've done is we've automatically analyzed the gel. We just get starting with the original image of the gel, and then of the 12 gels, split them up into individual gels, and then run through them. Yeah. So you integrate under the red box. Yes.
Because what about the area outside of dark bands, that's lighter colors?
Yeah. So I mean, you can do this in different ways. I mean, you can do it either like we did it last time, that we subtracted a general background, and that one can build into here also, and we'll talk about that a little later. It's sometimes easier to see what the background is then, and sometimes it's more difficult. So now we'll move on to another example. So this is a method that was developed to quantify how well antibodies work. So what we have here are beads that are coated with protein A. And then we make an antibody, and then we make the antigen protein and fluorescently label the protein. So, and then we bind the antibody, antibody binds to the protein A, and the antigen binds with the fluorescent labeled antigen binds to the antibody. So what we get is, if we get good binding between the protein and the antibody, then we get a strong signal. And as you see here, when we have the signal here, we see this strong rim effect. And that's mainly because what we have is we have a slightly transparent bead, and we have the surface covered in fluorescence. So then when we project this down into 2 dimensions, we'll have much more intensity from the rim than from the middle. But the noise is simply the beads. So that's sort of the bulk effect. We see the shapes of the beads, and there's no rim effect. So now this is just a calibration experiment where they added different amounts of antibody. So as we go from here, the amount of antibody on the beads increases, and we see that we get stronger and stronger signal. And then this is just the background and with a antibody that's specific to something that's not present. So we just get the non-specific binding effect here. So what we want to get out of this is a curve like this, where the control always ends up at 0, independent of the amount of antibody. And in the experiment, we want a linear relationship between the actual amount and the amount that we measure. So this is our goal here. So now we're going to go through a series of steps to try to get at least close to that goal. So the first thing what we can do is simply, so this is our goal here. If we just take the average pixel intensity in the image, a very straightforward and simple thing to do, then what we see is that we get a high background first of all, so that the control measurements are up here instead of down at 0. But we get a somewhat linear behavior on the measurements. So now what we have, of course, is that there's only about a factor of 2 ratio between these 2 points, which is not so good. So we're going to continue trying to look at how we can get this better. So one thing, if we compare just one of the experiments to one of the controls here, and plot an histogram of the pixel intensity. Then we see that for the control there is a large peak at low pixel intensities, and then there is a tail, and the experiment has a much longer tail up onto, which is not so unexpected. But there's not a really simple way to separate these. They overlap quite a lot. So we have, but one thing that we can observe is that the intensity doesn't go down to 0. There is an offset here of intensities. So we should be able to, by subtracting this, we should be able to get slightly better, a slightly lower background. So, and again, if we look at all the different images here, they all exhibit a similar behavior, that in the experiment we get a higher tail to higher intensities, and then there is this offset for all of them. So if you subtract that offset, so if this was when we didn't subtract the offset, just took the average intensity, and then if we subtract the offset, so we go here from a factor of 2 between these points, now we have a factor of 4. So it's getting better, but we still have a way to go. But on the other hand, so far we haven't done much. We've really just taken the average of all the pixel, the intensity of all the pixels, and subtracted the minimum intensity. So it's a very simple thing to do. But we can do a little bit better. So then, to do this, the next step that we can do is to just go take a slice through the image, and now come back to instead of looking at an image, we now look at the one-dimensional signal again. And in this case, we see very similar things. We see these thin peaks that correspond to the rims. When we go through the rims at the perpendicular angle. When we go along the rims, we get these wider peaks. And the similar thing for all of them. So we see that if we go through in the middle, we have these thin peaks that grow, and for the experiment. And then for the background, we have these wider humps that correspond to the size of the beads. So now, what we have to do now is sort of remove these wide peaks and keep the thin peaks. And you've come across that before. Anyone remembers? Sorry? That also, but more, I was thinking about the homework. So what we looked at last time was we had a wide peak in the middle and thin peaks on the side. And the homework was to find a smooth this in a way so that one would remove this wide peak in the middle while keeping the thinner peaks. And that's what so here in this case we have this smoothed curve, and then if we subtract that out, we have the thin curves left, and we can easily put a threshold. So we're going to exactly apply this technique to these images of these beads. So we have, if we look at the same slice again, we have the experiment with the thin peaks, and we have this the background. So now if we find the right smoothing, the smoothed curve will follow the control experiment and as well as it can, and then it will for the experiment here, it will not go into the thin peaks. So we will still be left with the thin peaks when subtracting that background. And as you see here, this just shows that for it works for all these concentrations, that we remove the larger peaks, and we remove the slowly the wider peaks here and the background also. So we can then do the subtraction and end up with a case where we in our experiment are left with our small thin peaks corresponding to the rims, while in the case of the control we only have a little bit of noise left. So you do this for all of the slices. So this is just showing it for one of them, but in this case we did it for all of them. And so this is the result then. So we did it two different ways. Either you just subtract the background and then take again the average pixel intensity. And now we're really getting close to 0 here. And if we add a small threshold of the optimal one, in this case seemed to be around 3%, then we really get the background down very close to 0. And we have now a factor of 40 between this point and that point. So we get a very good discrimination in this case. And this is, as you pointed out, then averaging over all the possible slices. In this case, I think 960 or so. So what we do is, so we take the max for each of these. We take the maximum intensity. So in this case, it would be around 400, let's say, for this one. And so we take 3% of that and set a cutoff there. So anything in this image that's after we subtracted the background that's smaller than 3% of 400 will be disregarded, and it's only the peaks that are higher that will be included. So yeah, so this is just summarizing what we can do with if we just look at 0 or 1 dimensions and do and so far we haven't done really proper image analysis yet. So our goal is that we should have the background always be 0. The experiment should be linear with the actual amount that we added. And this line should cross at 0. So the first step was to take the average pixel intensity. Then we get a high background. If we subtract the minimum intensity, we get slightly smaller background. If we do one-dimensional analysis of slices and remove the background, we get very close to what we're looking for. Now, this is of course not perfect. It's still a little bit of background here, and the more we can remove that, the better. And also the other thing is that there seems to be a little bit of a saturation. It's difficult to tell because there's so few points, but probably we remove a little bit too much and underestimate at high amounts. But what we then can do is to go back and do this analysis as a proper two-dimensional image analysis instead. And again, so what we have to, but it will be the same considerations that we have these, we see the entire balls as entire beads as sort of the background, and then we see the signal as these rim effects. So what we then want to do is to distinguish between this, as we've seen in the one-dimensional example. So a few steps that one can do, and it's pretty easy to do. So we haven't put those scripts up yet on the website, but in a few hours I'll put up all these scripts that generates these images. And so one thing that we can do is, the first thing we want to do is to do thresholding, to decide what is background and what is the foreground. So there are many different ways to do it, but the main thing is that you make a histogram of the intensities, and then you decide, you put you put a threshold to decide what is foreground and what is background. And in some cases this is quite straightforward, in others not so straightforward. So one way that that was developed by Otsu is to minimize the variance among these two classes. So minimize the variance of the parts of the image that's considered foreground and the parts of the image that's considered background. So if we do this here, now we apply a constant threshold first. That's based on this method that minimizes variance. We see that we find the rims a lot of times. In areas like this, it gets a little bit complicated and it doesn't look so good. So actually the better way here is not to use a constant threshold but an adaptive threshold that changes over the image and just uses the local regions to decide what's the image. And if we do that, we get an image like this where we see the rims. There's a little bit of noise here, but we get most of the rims pretty well identified. And then we're going to go through a few more steps on how to remove the noise. And one is erosion. And here what we do is we remove the white points that are have several black points in their environment. And one can set up depend on what geometry to use and what threshold to use to erode away. But now we see that we get nicely defined circles around the rim of each of the beads. So the next step, because there are lots of small objects and they probably introduce a bit of noise here. So one thing we could decide on is to focus on the larger object. So, and there we can easily remove the smaller objects, and that's in this case cleans it up a little bit. But again, it's none of these things are perfect. We see that we still have some arcs here, and some that are left over. But that's something that we can later on apply other methods to.
But aren't you using
Potentially, but depends on the application. It's always a trade-off that sometimes, I mean, one thing is sometimes you're interested in just, you know that there is a certain size of objects that you're interested in. But other times, in this case, we should probably try to not remove the small objects, or remove and compare. That would be the best thing to do. So then other things that we could do is to, for example, fill all the objects that are where there is a black area that's completely enclosed by a white area. So that's this object filling works that way. So as you see, it didn't fill this one because there is a little gap here, and wherever there is a gap, it doesn't fill it. So then we would have to apply other methods to maybe first fill small gaps before filling objects, if we want to get all of them included. So then when we have these objects, we often want to separate them and label each of them. And as you see in this case, several of them are, they touch each other or they overlap. So we want to be able to separate out the different objects. So there is a method called Watershed Segmentation where you can separate out the different objects. And the way this works is that you in your image the bright areas are the ones that are high, so that the topology that it goes up. But for this, what you do is you switch that around so that the maxima become minima. So you have these basins then instead. So this is if this is two peaks, you turn them around, and then you have these two minima. And what you do then is to start filling this with water, and then you see where the watershed line is between objects. So the way, so you find all the maxima. In this case, they're indicated here by arrows. So this just shows the mask as a binary image, and this shows now the topology, so that the red is the maxima. So then what you do is you reverse it, and then you apply the watershed algorithm to find where the borders are between different objects. And finally, you can then exactly find these lines, even in this case when these objects touch. But there is a part here that's a little bit lower in intensity. So then another example so this is a super resolution microscopy image that looking at two different proteins, and looking also seeing how they overlap. So we have two different proteins. So we have one of them is marked in green, the other one in magenta. So the first thing we start with is to separate the two images to be able to work with them separately. So we take the red image and the green image and separate them out. So we have the blue image we can throw away because there's no extra information here, because it's exactly the same as the red. So we have then these, we split the colors, And the next thing we have to decide is to what is background and what is foreground. So in this case, we can actually apply a simple threshold because we have our background is very nice and homogeneous. So this is, here we have the red and the green image separately, and we have the original image and the thresholded. So now they don't look so different probably, but we can zoom in and see that in this case we have a grayscale. So we have different intensities, and in the thresholded image we have it either black or white. So now we have our particles, and we can then apply another Python routine to label them. So what we do is we have this mask, and then every pixel, we either, when we do the thresholding, we either tell it, set it to that it's 0, meaning that it's background, or 1, that it's foreground. And then, but now the next step, the labeling step, is to go through and count how many objects there are. So we see that these 7 pixels that are marked 1, they are 1 object because they're all surrounded by zeros. Then we have another object here that is connected. Then the question is always, how do we decide what is connected or not? So then there are different ways. You can either say that it has to be on all the sides connected. So if it would be just several pixels here that were extended, and then another object that would be either divided into 2 objects, and so on. So that's, but there are different ways to do that, and it's up to the user really to decide what's the right way. So then, if we have the original, we compare now the original image, we have found our object, so we can just show the outlines of the different objects. And here, if we zoom in, we see that we have the green objects and the red objects outlined. And sometimes there is an overlap between them. So now that we have our objects, we can of course do all sorts of measurements on them. So we can measure their area and then look at the distribution of that area. We can measure where they're located, of course, what's the centroid, what's their eccentricity, what's their perimeter, and all these things can. It's just after we found the objects, it's a simple function call to get all this information. And then, since we have 2 different particles, we can look at how they're overlapping. And in this case, we, of course, can look at overlap from different points of view. So one, let's say if we look at the green, from the point of view of the green, then what the overlap is. And then we can then, by looking at these overlaps, build up more complex structures. And so, for example, one pair of structures that Dylan found in his work was what he called caterpillars and butterflies. So the caterpillars are the ones where you have the green filament, and then there is the magenta particle sits at one end. And the butterflies, it's when there are 2 fibers, and then there is a magenta particle in the middle. Then, of course, it's quite straightforward to generate thousands or tens of thousands of images, but it's not so straightforward to manually look through them. One thing that one would want to do is to be able to classify automatically into different categories. And this is just one example. I mean, of course, in different projects you will have different kinds of structures that you see, but in either case you would want to classify the structures that you're interested in and automatically find them. So in this case, it was what we did was to look at, again, we looked along the long axis of the green fibers, and again extracted a one-dimensional signal. So that and looked at where the intensity of the green along the axis, and then the intensity of the magenta particle and their overlap. Then, so this is for the butterfly, where the magenta signal is well in the middle. And then we have the case of a caterpillar where there is an overlap, but the magenta particle is definitely to the side at the end of the fiber. So again, it's definitely worth switching between looking at images and analyzing them, and then sometimes taking slices through them to get a one-dimensional signal that in a lot of cases could be easier to interpret. So then another thing, when we see these images and we measure different overlaps, we always want to know, is this are these two particles just overlapping at random, or is there some real structure there? So it's not always obvious. I mean, we see these lot of particles. Sometimes they overlap, sometimes they're close to each other. So one way to approach this is to do simulations. And so what we then can do is to take the measurements from the experiment, and then build a simple model. So I'll go through this model here with you. So the simple model is that we have these green and magenta ellipses that are bounded by a box. So that's corresponding to these stretches of the cell surface here, where we have most of the magenta particles. So we're just simulating that part. So what we do is first assumption is that the particles are ellipses, which is a slight simplification, but it's not that far from the truth. Then from the experiment we take how many green and magenta ellipses there are, and what their size distribution is, and what their circularity is. So those are the input that we get, and then we just randomly pick sizes and shapes of ellipses from these experimental distributions, and we randomly put them in the box, and also their orientation is random. And then we calculate the overlap and compare it to the experiment. And in this case, what we saw was that we got a much smaller overlap, even when we changed the box size and made it much smaller than what was actually, but we made it both much larger and much smaller than what was in the experiment. So we spanned a bigger parameter range. So, but this is a general method that you could apply to any image of two proteins. So then one can of course have more colors, so this is an image with 3 colors, and soon they will be able to have 5, 6 colors, I've been told. So there's but in general, the same methods can be applied to even if you have a lot of different colors. Then we can also have more dimensions. So this is a three-dimensional where we rotate. So we see that now, last lecture we looked at one-dimensional signals. Now this time two-dimensional, and then there is also the possibility of doing three-dimensional signals. And we could even have more dimensions. If we have a time axis, then we can have 4 dimensions, or we can do all sorts of other measurements. So in principle, we can have many different dimensions. And the methods that have been developed for signal processing do extend very nicely into many dimensions. So I hope I've been able to give you a little bit of an introduction to what is possible to do in image analysis. How one can just cut slices through the image to look at it in more detail as a one-dimensional signal. How one can do either global thresholding or a more local thresholding. How one can find and characterize objects, classify structures, and also test if there is some interesting biology or if it's just a random distribution that one sees. So next time we're going to talk about experimental design and mainly talk about how easy it is to get things wrong. So if you have any examples of experimental designs that are really bad, please tell us about them next time. Thank you.